Okay, so good afternoon, everyone. Let's start off the session with the third lecture by Silvia.
Thank you very much. I'll try to fight against the sleep that is coming upon all of us. So far, okay, we have seen mostly, I have dealt mostly with life cycles where you start from populations of scattered cells, you form groups, and this cycle repeats over and over. You get new groups, and you start over and again. And as I said, this is interesting because there is not very efficient way of purging diversity, even if it's detrimental in this kind of systems. But this afternoon, I will build on what Wenning has been discussing this morning to show you a couple of models that deal with this kind of situation. That is where groups beget groups. In reality, in many biological systems, you will find something that is in between, but this is the kind of setting that you can enforce somehow in the lab, as Wenning has shown. What is it relevant for, apart from the lab? Well, I think, for instance, of our microbiome. So the microbiome is inherited to a large extent from the mother to the child. And therefore, it has a genealogy that is somehow imposed by the fact that it can only live inside a host. And this genealogy is actually seemingly quite effective, even though it is always possible that there is some amount of horizontal transfer between different microbiota. The evidence that this vertical transmission is important comes, for instance, from some study of phylogenies of microbes of the microbiota that seem to be correlated to the phylogeny of their hosts. So there seems to be something going on between the host and the bacteria of the microbiota that makes so that certain bacteria, certain species are selected in certain hosts and not in others. In an experimental setting where we can sort of choose what groups or populations we want to keep and which ones we want to discard, it is relatively easy to show that you can end up selecting properties that are actually detrimental for the population if it was not subdivided into groups. This is exactly the example of Wenning, where you had to choose between darker colored or lighter colored units. So the possibility of having this efficacy in selecting these properties that are good for the community or the group is actually relies on the existence of this sort of ecology that keeps groups separated and therefore creates a lineage of groups. So this is what Paul Rainey and colleagues call ecological scaffolding. And there can be many different ways of scaffolding ecology, but what we will do today is just sticking to the kind of so growth and dilution experiments as Wenning explained very well. So the idea is exactly what she was talking about, so I will sort of recap some key features. So let's say that you can, and nowadays this is becoming actually technologically possible, you can keep a community that is composed of many species of microbes, for instance of bacteria, within a droplet. This droplet will be, even though it's tiny, it will still allow to keep a large amount of bacteria. And now you let these droplets flow in front of some kind of detector, and perhaps you have you have inserted in this community one bacterium that is fluorescent, and you want to select for higher fluorescence. For instance, the bacterium who is, I don't know, degrading cellulose, and then you can pick out what are the droplets that contain a higher concentration of this. So this is not operational yet, but I think people are getting there. In the absence of such a high throughput technique, one can also do it in the lab with test tubes. So just start with a number of different communities. For instance, you could, people just sample from the environment and then, you know, randomly dilute things down, have some inoculum. These inocula are, you know, controllable in diversity to a certain extent. And then you incubate these cultures, let the different species grow, and then you will assess in these adult communities some trait that is a trait at the community level. They are not mixed. This is just a growth phase. So you put them in the tubes, you grow them up. And then you look at the end, what is the feature of this community, of this adult community. So species are interacting, but each one is interacting in its own, you know, ecological setting. So if you measure some property of these different communities, in general, you expect to see some kind of distribution, and then by some criterion of choice, you will select the communities that maximize a certain property. And once you have selected these best communities, then you need to go through a step of reproduction. So this is, the first step is growth, selection, and then you can reproduce by picking out, so these communities, redilute them. So there are many different ways to do this. You can dilute, just sample. You can just transmit what it is. You could introduce at this point some degree of immigration. So to make things simpler, just let's imagine that each parent, each adult community that belongs to this tail will be the parent of some of the newborn communities of the following collective generation, so the community generation. And in general, you will have some mutations that occur in this time. So both in the growth phase and you could artificially introduce them at this phase by adding some migration. But in the models, I would consider this will all be lumped into this reproduction phase. So at the moment of reproduction, there will be some mutation that might have been acquired at different times during growth phase. Is this clear? Okay. So then the question is, of course, can we select something that goes against the traits that are favored by natural selection when you keep all communities mixed? So one of the first attempts to implement experimentally this idea that groups can be selected and that this group selection can be effective actually to get an improvement of some group level quality comes actually not from microbes but from hens. So this influential paper from the Journal of Poultry Science. So what Muir did is that he had hens in boxes, and each box had, I forgot whether 4 or 5 hens. So these hens can be either selected individually to produce some more eggs. So you can, instead of keeping them in a cage, you just pick the best hen, the one that produce more eggs, and then keep going. And of course, as you would expect, this is normal, artificial selection. So you eventually get a hen that is producing more eggs. Or the alternative treatment was to put them in cages. These cages originally, at the beginning, they will have less egg production than the sum of individual hens, or if isolated. And then over time, the egg production of the best cages will improve progressively, and the cage will produce in the end as much eggs as the sum of 5 hens among the best selected individuals. So perhaps you say, okay, this is not a great advantage, but the interesting thing that he observed is that if you now put these 5 best hens in a cage, they will produce nothing, because they are super aggressive. These are, the best hens. Those who produce more eggs are basically incapable of working in a group setting, whereas those that have been selected at the group level, they are capable of living together, and in the end, they provide the same amount of eggs as the other ones. So if it works with hens, then it should work with microbes. And people have tried, indeed, to do the same thing with microbes. The idea here, that there are actually other experiments that have been performed with, what's the name of the bugs? Insects, with insects by Goodnight and colleagues. So this is an example for microbial communities. The reasoning is that the health of a plant, as measured, for instance, by the biomass of this plant, or by, I don't know, how green its leaves are, is connected to its microbiome. So if you start with a number of plants that have the same microbiome, and now you keep selecting the microbiome associated to the best plants, so the plants, for instance, with higher biomass, and then you propagate this, then in the end you should have a microbiome that is actually providing what a plant needs to increase its biomass. So the results are not exceptionally striking, but you can see that there is a difference between the ancestral microbiome and the 2 evolved microbiomes. So one is the microbiome that has been evolved for lower biomass. So the selective pressure has been put into lower biomass. The other one has been put into higher biomass. So there seems to be an effect of this group level selection. It turns out that among the different experiments that have been done, so results are a bit contrasting. Sometimes you get indeed improved collective function. You improve the function you are selecting for. Sometimes you don't. So it seems that some theoretical work is needed to understand under which condition would you expect this function to improve. And as Wenning was pointing out, there are plenty of parameters one can change in these kind of experiments. Even already with 2 species, you have many. You can imagine if you have a whole microbiome that has hundreds of species. So how do we decide what is the best way to obtain an effective selection for a collective property? And this will be the subject of this talk. So before I go to the more evolutionary part, I would like to stress that there is some, I'm not sure whether to call it misunderstanding, or at least there is some semantic differences in what people mean by community evolution. And this is because you can get changes in communities that are composed by the same species, by the sheer fact that you have organized them in this lineage of communities. So imagine you start with a certain composition of your community, then you let these different species grow, you run community function, then you see the new generation, you keep going, and then you will have a changing community composition over time. Notice that here you are not changing species, so species do not need to evolve. They can stay the same. Still, depending on how you do these different steps, you can get different outcomes of what will be the final composition. So the final composition is not just one. It depends on many different things. In simple cases, it will be just one, but in general, it will not. So people, yeah, like this was called directed evolution. I usually don't call this evolution. Yeah. 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 I guess, yes, you would need to wait a long time to get a community that is very diverse. Something, yes, I didn't stress probably enough is that there are certain community level features that are really directly related to biodiversity. And this has been studied a lot in many different communities, like primary productivity, total biomass, resilience to stress. So all this actually require the existence of a number of different species. If you want to obtain all different species starting from one, it will take likely a very long time. So somehow the yeah, there is a functional complementarity in many communities that perform certain particularly complicated biochemical transformations. Exactly. So the kind of functions we are interested in here is not something that can be performed by one species alone, but something that is the outcome of the interactions of 2, 3, or many species. Okay, so this is, yeah, despite some people call this evolution, I would rather call this like ecology of communities that are embedded in this multi-level structure, so in a nested kind of genealogy. Yeah. So in that sense, it is, right? Like if you yes, but you don't really have, yeah. No, I mean. I know. It's a personal. Let's say. I think it's useful to make this distinction because one thing is whether you have a mechanism to generate novelty in the traits that are present in the community, and another one is not having it. So this makes this kind of models particularly difficult to explain because there are different timescales that are associated with different processes and different levels of description. So I try to start very slowly with a two-species community and try to explain what are the different problems. Then when it will go on tomorrow. Anyway, yeah. So just talking about mutations. So you could imagine that while you're doing these experiments, there will be some mutations that happen within the strains that you are using, or the species you are using. However, you will only see the effect of those mutations at the adult level, because that's when you are assessing the function of the community. So this poses a bit of problems on how do you model this. Do you need to model really every single event of mutation, or can you actually model mutations as a bulk based on their effect at the adult stage? I keep thinking that this is really analogous to what happens to genes within cells. So within the lifetime of a cell, you might have multiple mutations, but in the end, what really matters is when the cell divides, what will be the reproductive advantage or disadvantage that is created by the set of mutations it had during its lifetime. So it's a bit the same here. So I will discuss a bit this later, because this has consequences also on the way we conceive how evolution can proceed. And of course, there are many sources of stochasticity here. You can have stochasticity in birth-death processes at the level of cells or species. And this, if you have a sufficient amount of cells to start with, you can sort of neglect and think that everything is deterministic. Then you have some important stochasticity in the moment in which you are reproducing. If you are diluting down enough, then you end up having an inoculum that is made of a small number of cells. Potentially you lose some species in doing this. And then you can introduce stochasticity at will in the process of selection. So since there are all these different dimensions, my models will be mostly deterministic, and I will put in stochasticity in a way that is not too disturbing. But one can do numerical simulations to check that these deterministic approximations make sense. Okay, so since it is actually hard to make this kind of experiments, people have started experimenting with numerical simulations. So you have virtual communities that are described, for instance, by multi-species Lotka-Volterra equations. These equations describe the variation in time of the abundance of each species as a consequence of the abundance of the species itself and the abundance of the other species. And so in its simplest form, one just takes into account pairwise interactions. So the interaction of one species with the others is the sum of the interaction with one species at a time. So if you simulate this kind of nested populations, and you are trying to reach a collective goal, in general, you will. So the prediction is that from these models is that you will always improve your function that you are selecting for, whereas if you choose randomly, you will not. Okay, this is some kind of drift, but drift of a collective property of the community, and this is like directional selection. More or less same conclusions have been drawn in models that are a bit more realistic. So Lotka-Volterra equations do not explicitly say what is the basis of competition, for instance, between different species. Then people can use resource consumer models where you have a number of species and then a number of resources. Species tend to use the same resources and therefore they compete for them. These are, you have more variables but less parameters, so it's a bit of a matter of taste whether people choose one or the other. So in this case as well, you have a treatment where you are selecting for a high value of some variable that is just some function of the abundances of the community. And you see that you are selecting, what you are selecting keeps this property until you stop selecting, and then will drift back to something that corresponds to the case where you have not imposed selection. And if you are actually selecting for something that is against, so for low values of that property, then you will have, basically, you will go further. This is a distance from the goal. And again, you will drift back to this neutral case when you discontinue selection. Okay, all this is, goes in a certain way in the direction of what you would intuit, but it has a lot of parameters, and it's very hard to generalize, so one ends up basically doing the same work as you would do in an experiment. You take, your selected communities, and then you try to find patterns of what changed, what did not change. Sometimes you find that actually it was one single species that was able to adapt to the goal. Sometimes multiple species are necessary. So it becomes almost naturalistic observation of a community simulation. So this is why people are trying to find ways to simplify down the system. So some interesting ideas come from the sort of theoretical framework whereby if you try out many compositions of a community, and composition might mean in certain cases what species are present, and in some other cases it means what species are present in what proportion. So both are called composition, unfortunately. But let's say, so you have a state of a community, and then let's imagine that you are measuring from this composition a certain function. And then you could imagine that this function is actually, so you are defining a landscape. And one might imagine that evolution will actually be a gradient climbing process in this landscape. And of course, gradient climbing can be efficient or not, depending on what the landscape is. So if the landscape is sufficiently smooth, then it will actually allow you to improve your function. But if it is very rugged, then you risk of getting stuck in local minima, and then you will need some other ways of improving diversity or getting out of those minima. So there is quite some work that is done in this moment, and it seems that for at least for the kind of communities that are mostly studied in the lab right now, this landscape appears to be relatively smooth, so there is some hope of finding some lower dimensional descriptions of this ecological process. All right, now I'll go to the model. If somebody has a question at this point, yeah. Yeah.
When you say selection, how do they model it? Is it like a multiplier of the total growth rate or something?
No, they do exactly what I was saying. So they run the simulation. Typically, they wait until the system is equilibrated, but they might not sometimes. And then you just score many different communities, simulated communities. You get those who get the highest score, and then you reproduce them in one way or another, typically by diluting or by copying. Sometimes adding, so if it's evolution, then you add some new species, for instance. And then you keep going. So I hope that this will be more clear with the case of two-species communities. So I just wanted to mention that there are several models out there that have considered two-species communities. So these are the models that Wenning started discussing this morning, so I will not add anything. And this is another interesting model for microbiomes that includes both vertical and horizontal transfer of species. And that concludes that actually multi-level selection is not always effective. It depends on the parameters of the system, as one could imagine. And in some cases, it is within host selection that wins over between host selection. Okay, I will present the model that was developed mostly by Guillaume Doucier and is a collaboration with Amaury Lambert and Paul Rainey. And this model consists in considering, so these droplets, if you want, these mini communities, communities made of just two species, so blue and red, and it's the same kind of convention that I used yesterday. So once you have a given proportion of blue and red, then you will associate to this a color of the community as a whole. So this color can go from completely red to completely blue. And let's say, for this case, which is different from the public good game that I considered yesterday, let's consider that what we want is to have a purple color. So we want to have a community that has half red and half blue. And this is the optimal because they have some complementary skills. So we will eliminate some communities, just the most extreme phenotypes. So I will speak about collective phenotype or community phenotype. That is the color of these communities. And then propagate this through some, actually through sampling. In this case, we just take a certain number of cells out of this adult community. This is our newborn generation. We will let this grow and then score this again, select again, and keep going. So let me be more precise on what we are doing. So reproduction by dilution means that we fix the bottleneck size. So we fix, actually, I could have chosen a better example. There should be the same number of cells in both cases. I apologize. So the bottleneck size of 4, you should have 4 cells in both communities. So this is a parameter that when it's large, basically the composition of the newborn will be identical to that of the parent. But as it becomes small, then you will have a certain distribution of cells, of colors in the newborns. Then we will allow particles to mutate. So just one mutation will be possible at every round. So let's say the red color can change to another red color, and blue will change to another blue. That means that they will retain the property of being either red or blue, but they will be different, and they will be different in their traits. So the traits that are attributed to each of the colors are basically the traits of growth rate, just of the color when it's alone, and the traits of interaction with other colors. So the bottleneck, so you have, like in a test tube, a given community, and then you are diluting this down. But in this dilution, instead of fixing the amount at which you are diluting, you fix how many cells you will put in the next generation. Let's say I have 10,000 cells to start with the parent. The offspring will contain 10 cells. And then I have to sample 10 cells out of these 1,000 cells. And then since you sample 10 cells, the composition will not be exactly the same as the parental composition. Of course, the smallest is the bottleneck size, the largest is the fluctuation in the composition of the newborn generation.
It has nothing to do with, usually we say bottleneck when certain
Well, when the population size shrinks, so it's the same. The only thing is that I'm imposing it. I'm just saying, okay, so I shrink it artificially. All of a sudden, I take out this number of cells. And then once I have this newborn generation, I will let it grow, and the growth will occur according to these particle traits. Okay? So particle traits, I forgot to write it. So there are growth rate, intraspecific interaction rate, and interspecific interaction rate for each of the colors. Okay, so we have 6 parameters in general. In order to make things simpler, we will keep fixed the intraspecific interaction, that is the carrying capacity of each of the colors, and we will let evolve the maximal growth rate and the interspecific growth rate. Okay, and the question is, will purple color be maintained in the population, and how does this happen? So let me tell you a bit more what happens between generations, so between the newborn and the adult stage. So let's say you start with a given number of red and blue cells. This is a point in this plane. And now this is the Lotka-Volterra dynamics that we have chosen. So here you have the 2 maximal growth rates of the 2 species, and these are the intra and interspecific competition rates. So in general, this will define a trajectory in this plane. But this trajectory will not go necessarily all the way to its equilibrium, because after a certain time, I forgot whether I mentioned this, no? But so this growth happens for a finite time that I will call big T. So after a certain time, we will get to some point, and that will be the adult state. And now we dilute down, and dilution in this plane just means that you start from here and you draw a line between this point and 0, and the intersection between this line and this line, that is where the 2 numbers sum up to the bottleneck size, will be the state of the newborn in the following generation. Okay, this is without mutations. This is just ecology. Is that clear? Okay, and then you can keep going. And if you keep doing this over multiple generations, you will end up having, some state that is stable. And notice that this state does not need to be the same equilibrium as the system when it is as this system. So this system, we can compute the equilibrium. It's easy. But this green state will somehow belong to a transient. And it's actually very hard. We tried to do something analytical about this, but even with the Lotka-Volterra system, that is a simple system, is not that easy to demonstrate that you have convergence to a given equilibrium in this system. Okay, but we see this numerically, it always converges to some equilibrium. So what point exactly it will converge to will depend on the parameters that the experimenter can modify. So the duration of the growth phase, you see that if this was much shorter, then you might go somewhere else. And the bottleneck size. Now, we would like to represent this ecological dynamics by a discrete time process that is that is connecting the adult state in one generation to the adult state in another generation. Okay, this is again ecology, so no mutation so far. And in order to do this, we need to know if we start with a newborn with a given composition, what will be the composition of the adult? And in order to know this, so this is not something that is easy to compute analytically, but you can do it numerically. So you just change the initial composition of the newborn, and then you plot this line. And this line is what we call the growth function. So it is just telling you what will be the adult given the newborn. Since everything is deterministic, this is a nice line. And then what happens is that you can compute what is the ecological trajectory starting from this line. So you start from here, 50%, then red will have an advantage. Then you project this on the diagonal. The next generation, you will start higher, let's say 0.7%, and then you go up, and exactly as I showed before. So in this case, you will have that in the end, you will have just one color in the system. Okay, if you add some noise, for instance, here in the newborn composition, this will still tell you what will be the adult composition, as long as stochasticity is not too important within generation dynamics. So now going to the question of will we be able to maintain the purple color? Now let's imagine that you have, this is a representation of the genealogy of the different collectives. So you start with some collectives that have a composition that is about 50%, okay, so the percentage of red particles. And now you apply this dilution and growth dynamics over and again without adding selection. So you just randomly choose what are the collectives that will survive to the next generation. Actually, you don't even choose in this case, you just keep all of them. No, I think we randomly choose so they can die and be substituted by the offsprings of some other collective. So what do you, well, I'm not going to ask what do you expect because you can see it, but can you explain to me what's going on here? So I remind you that the blue and red are not identical. They have different parameters. So what kind of collectives will I see in the end? What will be the color of the collectives that will be remaining in the population? Yeah. Yeah. So, and what will determine if it's blue or red? So the monochromatic, they will all be monochromatic in the end. So monochromatic collectives are absorbing states. Once you are in just one single color, you cannot get back particles of the other color. So you will necessarily, if you hit all blue or all red, you will stay there. So what will determine whether you will be blue or red in the end? So you start with something that is composed of a mixture. So will it be blue, red, more blue, more red? Initial conditions are fluctuations around 50%. But the two species are not the same. Just as an intuition, for instance, let's say that one species is growing faster than the other. So if the red species grows faster, then will it be more likely for it to be fixated or not? Opinions? Who votes for yes? Right. Yes, it will be more likely to fixate because actually at every round there will be more and more red cells. But still, you can have some stochasticity by which if here the bottleneck size is small, so you might have that at the beginning you end up having more blue cells. So it is possible that at the beginning you just get blue cells. So we'll have some rare cases they will go up to blue. Here you don't really see it. Actually, I think that this is the contrary. In this case, actually, it is blue who grows faster than red. So it's easier to go up than to go down. Okay. Now let's imagine that always we don't have mutation. Yes. The light blue line is the average phenotype in the population. So you take the composition of all your collectives and you make the average. So the average will be biased toward blue, because there will be more blue than red in the end. So now let's imagine that we switch off selection. So we select those collectives that are closer to 50%. But we do not let species evolve. So the parameters of blue and red keep remaining fixed. What would happen? Will we be able to maintain the purple color or not? So, who votes for yes? Who votes for no? Right. Okay, it turns out that actually you can. You can, thanks, provided you have a sufficiently small bottleneck size, thanks to what Eurosasmari has called the stochastic corrector. So that means that even if you have some small differences initially in the growth rates, you will by chance create some newborns that are biased in the opposite direction with respect to where the intra-collective competition pushes you. So if blue grows faster than red, you will always have some collective that actually have a bit more red than their parents. And selection can actually keep you going with this. You just keep selecting this few. I mean, you don't need to have many. It's enough to have one that is sufficiently biased towards red, and you can keep that one. You just keep one, and then you distribute it in the different collectives of the new generation. So not every parent has an offspring. You know, you will select a subset, and then you can see that this sort of umbrella graph tells you that single parents will have multiple offsprings. So you are correcting at every step. Stochasticity allows you to correct the composition. The downside of this is that if you stop selecting, then this will be gone. So you will be back in the previous situation, and then you will again lose the purple color. You will only get either blue or red. The other thing that is slightly annoying is that in all this population of collectives, there will be just a tiny fraction that has the right color. All the others will be distributed around. So it's not a very, very efficient method to actually select for this collective function. Nonetheless, you can. You also need a pretty high, pretty low bottleneck size in order for this stochasticity to be effective. If you are just transmitting, exactly the composition of the parent to the offspring, then this will not work. So there are pros and cons to this stochastic corrector model. And what I will show you is that everything changes if you now allow particles to evolve their parameters. So this is a simulation where we allow evolution to occur. So as you see, changes occur on a much, much longer time scale. And so you will have that the population of these collectives will progressively converge in phenotypic space towards 50%, that is the target. This is another representation of the same thing. This is the density, so the fraction of collectives in the population that have a composition that is close to 50%. And as you can see at the beginning, there are just a few. And then over time, you will have that most of them are very close to the target composition. So what is even cooler is that when you take away selection, this does not go away. So this is like a zoom in at the end of the simulation. So with respect to before, where at every generation you would have colors that were scattered around, here going from generation to generation, yes, it is enlarging a bit the distribution, but it tends to be really peaked on the target. So it takes a long, long time to lose this phenotype. So here we start with the selected community and took away selection. So now, as you can see, in the end, you will indeed lose this phenotype because now you have random mutations that can make different phenotypes appear in the population. But this is really drift. It's a long, slow process. So this is cool. That means that the kind of phenotype we have here gets inherited over multiple generations of the collective. That is what we wish if we want to select for a property. We don't want to select for a property where we have to keep selecting in order to keep it there. We want to select for a property that stays there after we stop selecting. The interesting thing of this model is that one can look at what happens to the parameters of the system along this evolutionary trajectory. So, for instance, the growth rates of the two types and the interaction rates. And there are actually several steps that occur for this during this selective process. So the first thing that happens is that in general you will have, actually I think it's, sorry, I skip a bit ahead, then I go back. It's easier to see it here. So this is a magnification of the first 30 generations. So in this moment, there is one type, the red type, that is strongly favored. So here, the two colors are just maintained by stochastic selection. What will happen is that both types will try to grow faster and faster. And they do so by aligning their growth at the beginning. So if growths are aligned, so if the two types have the same maximal growth rate and they're growing exponentially, then if you start with a certain proportion, you will have a certain proportion at the end. So this is like the safest way to get a maintenance of the proportion. Just we grow at the same rate. So they could stop there, but actually what happens is that they want to grow faster and faster. So they will develop a faster and faster growth rate, and as they grow faster and faster, they will not be anymore in the exponential phase by the end of the generation. They will start entering stationary phase. And what is stationary phase is actually the moment when interactions start to play out. And as I told you, so interactions can be different between the two species. So if you look at the growth function that I defined before, what you observe is that after this alignment of growth, you have that this function modifies so that you get an internal fixed point. And this internal fixed point is actually like quickly stable. So as soon as you have some stochasticity, you will still have phenotypes that are quite spread out, like here. And then slowly, this will evolve towards a growth function that one could see as a canalization of development. When this growth function has this shape, then somehow, actually I'll go back to this other slide. This is what happens in the end. You get a growth function like this. What this is telling you is that irrespective of what is the newborn composition, you are ending up in the same proportion of blue and red in the adult stage. So it is somehow correcting the initial randomness that is given by dilution. So you go from here to here. What is interesting is to look at what is the intrageneration dynamics that corresponds to these two cases. So at the beginning, as I was telling you, you have that the blue, so I think, I mean, in this I have switched the colors, I'm sorry. So it was always the blue color that has, I will correct this in the final slides. So it's the blue color who grows faster. And so at the beginning, you just have that this advantage of the blue color accumulates over time. And in the end, if you now, you do not put mutation, you just take the ecological dynamics of this, you will have that one color will grow faster. And this is, it is always one type. I'll tell you later why, which one. But in this case, the red color will grow faster. It will grow, and it will overshoot. And then it will realize, actually, the blue color is much less numerous than I am, and we need to be 50%. So these guys will actually start dying out by competition with the blue color. And in the end, after a time t, there will be 50%. So one color will evolve. So these are the growth rates, and you can see that certainly they increase, but then they undergo some kind of semi-random variation. They are not any longer all that relevant, because what is really relevant to define the final state are interactions at high density. And these are the interactions. So the blue type, at a certain point, just it keeps doing what it wants. It is ignoring the red type. The red type, on the other hand, is evolving a response to the blue type. So there is one type that becomes a master, if you want, and the one type that becomes a slave. There is this asymmetry that evolves. And what is interesting is that the type that is the master is the slowest growing type at the beginning. So, sorry, not slowest growing in terms of growth rate, but the type that has a lower carrying capacity. So the type that has a lower carrying capacity is the one who eventually decides where they are going. I think this is very interesting. There is some, I don't know whether this is total science fiction, but if you think about endosymbiosis, between the nucleus and a bacteria that is engulfed, then it would be the nucleus that is the one that eventually controls all. And the bacteria have to adapt to the time scale of the nucleus. This might be a bit far-stretched. Okay, I see I need to speed up. So you can actually compute what is a stable fixed point as a function of the parameters that can be controlled by the experimenter, the bottleneck size, the duration of the growth phase, and it turns out that you need to have a sufficiently long growth phase in order for coexistence to be selected. So if you are diluting too quickly, you never allow these two types to really start interacting. You keep things in exponential phase, and then just the type who grows faster will win. You also need a sufficiently large bottleneck size because you need some variation between newborns in order for selection to be effective. Okay, so this is perhaps a bit long to explain, but this is representing the ecological equilibrium given the parameters of interaction between the two species. And one can see that actually in this space, evolution will be a gradient climbing process that will bring first on this line where the two growth rates are aligned, and then it will push actually interactions to be more and more nice to one another. Actually, there will be an asymmetry if you don't allow them to become negative. So if they are negative, they are facilitative. If they are positive, they are competitive. We started with competitive communities, and actually these communities will be pushed towards being more and more mutualistic if you allow them to. And I will go back to this in a while. And then one can start asking, so what happens if actually instead of having 4 parameters that are evolving at the same time, I have some trade-offs between these parameters? And then the variation, for instance, of 2 parameters will depend on the variation of the other 2. And what we find is that you will still have this gradient climbing process, just the trajectory in this space will be modified. Yeah. This line is when the 2 growth rates are aligned. Instead of going here. Yeah. In this case, I don't know. Yeah, this goes perpendicular. That would be the most natural. I don't know. I have to think about it. I forgot exactly what were the details in the. I think it probably has to do with the 2 trade-offs are not exactly equivalent. So both trade-offs have the same shape, but since you have different parameters of the 2 species to start with, you will have that one trade-off is probably more limiting than the other. But I should check. Yeah, good question. So just to conclude with this very simple 2 species communities, so collective level selection is able to improve the inheritance of collective functions. Do you remember that the phenotype is maintained over several generations? Evolution of this function and its stability is associated to the evolution of the interactions. So species start to listen to one another. And the selection protocol influences the speed. This I didn't discuss much, but so the speed of collective adaptation depends on the parameters of the system. Okay, now in my last 20 minutes, I will try to tell you something about what can we do if instead of having 2 species, we have a large number of species, in principle, an infinite number of species. So microbial communities in the wild are composed of hundreds or thousands of different species. And of course, as you can imagine, the problem is much more difficult because there we already had 6 parameters for the interactions and growth rate, and we fixed 2 in order to get 4, and these 4 we could handle somehow. If now you have hundreds of species, even if you stick to a simple Lotka-Volterra kind of dynamics, you will have, S, if S is the number of species, you will have S times S parameters that are associated to intra and interspecific interactions, plus you will have S parameters that are associated to growth rate. So how can we get some kind of insight of what will happen? And we would like to have something that is sufficiently robust to believe that it's general, and we don't need every time to do a simulation to check what is the effect of this or that experimental parameter. So we tried to generalize what you have seen to the setting where within each of the communities you have multiple species, and each species can have different abundances. So the protocol is a bit of the same. So we start with a growth phase. So we start from, okay. So we go from an initial inoculum to an adult stage. What determines what will be the adult stage is the ecology, that I will precise what these are later on. So there is some ecological rules that tell you how the newborn is transformed into an adult. And then we can score these adults. Oh, sorry. This will depend on the initial condition. Of course, different communities will end up having different adult states. Then we will select these adults according to a target function. So the analytical results are valid actually for any target function T that is a function of the composition in terms of abundance, so species and abundance of the species of the community. But to make things simpler, think that this function is the total biomass. So we just sum up the abundances of all the species. And what we want to do is to maximize this biomass. So we select the communities that have the highest biomass. And now we reproduce them in a way that is much simpler than what we did before. We just copy and paste the adult state. So we assume that they reach an equilibrium, and then we copy this equilibrium to the new generation. And if we stop here, of course, nothing would happen. What we do is also to introduce some mutation. And these mutations will be introduced in the matrix of interactions between the different species. So in the end, we will have a number of mutated newborns, which will be different from the adults of the previous generation. And then we can let them go again to their ecological equilibrium and start again. So this was generation 1, this is generation 2. Here we select and start again. Yeah. No, I'll tell you, I will now specify each of these steps. So first of all, the ecology. So ecology, we will take what is called generalized Lotka-Volterra equations. So these are just exactly like the equation I showed you before. The only thing is that now you have many more parameters because you need to specify what is the pairwise competition between different species. At the beginning, to start with, let's say, I put the wrong matrix. Anyway, perhaps you can't recognize this, but we will choose a matrix that is initially randomly chosen. So we will assign randomly the entries of this matrix according to a given mean and a variance. So we fix mean and variance, and then we choose this randomly. Okay, this one is not random, and I apologize, I'll change this as well. Then we will also choose randomly the carrying capacities, but we will not change them. We will also set to 1 all the growth rates. And this is because since we are looking at the long-term solution, actually the growth rate doesn't matter because you are looking at the equilibria of this part of the equation. So this will influence the transient, but it will not influence the ecological stable state. So this is the number of species. So this is, for instance, what is the intensity of the competition between species 10 and species 30? This is how you read these matrices. Okay, at the beginning, we also choose matrices that do not have a specific symmetry. So the correlation coefficient, average correlation coefficient is 0.
The diagonal should have been the same, no?
Pardon?
Diagonal would be what, 1 over ki?
So the diagonal is set to 0 because we put out the interspecific competition here. But since these matrices can be arbitrarily large, actually the diagonal doesn't really matter. So this kind of system has been studied by statistical physicists who have drawn the phase space. And they found that depending on the statistics of this matrix that are chosen so that they scale with the number of species in a way that you always have that the total effect of all other species is more or less of the same order as the effect of one species on itself. Then this mu and sigma are measuring the average intensity of competition and the variance, the heterogeneity in these coefficients of interaction. So here, when mu is larger, then you have more competitive interactions. When mu is positive, then interactions are mutualistic on average. And what they found is that if heterogeneity is small enough, the solution of the ecology has one equilibrium that is globally stable. And then if you increase it further, you will go to chaotic behavior, but we want to stay away from that because it's a bit messy. Another interesting thing that you can obtain analytically is that you can compute the total population size, the total community size, as a function of mu and sigma. And this is the gradient that you see here. So the total community size is increasing as you go from more competition to less competition, and from less diversity to more diversity. As an initial condition, we choose to start from here. So we have a community that is initially competitive, and it is at equilibrium. And now we want to introduce some kind of mutation process in this. Okay, if you don't mutate, you just are stuck here, and that's it. Yeah.
Question on terminal. Of course, you wanted to stay away from chaotic things.
Yeah.
But usually, chaos is associated with deterministic dynamics.
Yeah.
Here, your matrix is random.
Yeah.
So isn't it a stochastic dynamics? And hence what
No. So the matrix is set once and for all. Just the entries are randomly chosen. So there's nothing stochastic here. So, but when we make this mutation on the interaction matrix, we want to do it in a way that is not biased. So let's imagine that we want to mutate a community, have a new community that is close to the old community, but it has some difference in the interaction rates. So the way to do this is to actually define this perturbation in a way that, on average, the effect of the change in the interaction matrix will provide you the same kind of phenotype, in this case, the total biomass of the community, as the parent. So you have a parent, you create many mutations, you will have a corresponding distribution of the total biomass. Now you want that this distribution is peaked in the biomass of the parent. So the process of mutation is not biasing the property that you are trying to select. Because otherwise, of course, if I am selecting, if I'm already generating variants that have higher biomass, then biomass will increase. That's pretty obvious. So by doing mutations in this way, then this is not the case. You will create a number of matrices that have different total biomass. And in this distribution, so you start with one parent. You create a lot of mutated offsprings. And then for each of these offsprings, you compute the total biomass. And the total biomass will be, so if this is the biomass of the parent, will be distributed around this average, okay? And the width of this distribution is measured by epsilon, by the parameter epsilon. So epsilon is sort of the mutational step. And the statistics of this matrix are such that we are not altering the cross correlations or the statistical properties of the matrix on average. Then among all these mutated communities, we would choose just one, that is the one that maximizes the target function. That is, formalized by that where the dot product with the vector 1 is maximal. And then we just keep going. Okay, this is just sort of a synthesis of what I explained so far. And now we want to see how does the matrix of interactions change over generations. So any guess? What will happen to this matrix? We start with a random matrix with an average value of competition and a variance. Will it become more variable, for instance? Do you expect the community to have interactions that are more variable or not? No? There's a no there. Someone says yes? Yeah. The phase diagram, exactly. Exactly. So you should decrease interaction rate and you should increase diversity. This is what happens. So you always manage to select for more and more abundant communities to the point that in the end they diverge, and this is sort of a side effect of Lotka-Volterra because you are selecting actually for interactions that are more and more mutualistic. And if you look at what happens to the statistics, you will have indeed that the average mu, so the average level of competition is decreasing, and the variance of the interactions is increasing, exactly as you would expect by analyzing this diagram. You mean this? Yeah, but gradient ascent will be more in like this. So the direct gradient ascent. But actually, it's just qualitatively the same. It's not quantitatively the same because if one now takes an evolved community and project it on the surface that is identified for random communities, it turns out actually this community is no longer random in the end. We start with a random community. Evolution pushes you in the same direction as random communities, but in the end, your community is no longer random. And then the question is, what is it then? What are the features, the statistical features of this evolved community? It turns out that as you go from this initial random matrix to this final matrix, you have selected for some structure in the interactions. And this is associated to a transition in the eigenvalues of the matrix. So if you compute the spectrum of the matrix, what happens is that at a certain point, you will have one isolated eigenvalue that comes out of this bulk of eigenvalues that is associated to the random matrix. There is also another eigenvalue up there at the beginning, that is this one. So this one is not present at the beginning. You only have one eigenvalue that is on this side, and you have this bulk. So this other eigenvalue pops out at a certain moment, and it is what provides the structure to this matrix. It has some interesting properties. So I skip all this. One can write down the equations for the, so one can write down how the matrix at time tau plus 1, at generation tau plus 1, depends on the generation at time tau. And this depends on a number of things. Among others, what is the distribution of the carrying capacities at the beginning? That is fixed. It depends on what is the selection target. And it also depends on some other features like the mutational step, as you could imagine, the number of species, and what is very interesting, it depends on the number of communities that you have, that you are subjecting to selection. So this is telling you that it is true that if you increase the number of communities, you will improve the speed of convergence to, the speed of evolution of the phenotype. But it goes like the square root of logarithm. So it doesn't make a lot of sense to go, from 10 to 10,000. Perhaps it is more important to go from 3 to 10. So perhaps, the main objective is not necessarily to parallelize in order to have a high throughput kind of system, but actually to be able to affect some other parameters of the system. Okay, and another interesting feature of this system is that the eigenvector that is associated to the eigenvalue that has popped out is actually highly correlated both to the distribution of abundances of the species and to the carrying capacity. This means that the species who become more mutualistic in the end are actually those that at the beginning have a higher carrying capacity, which is also interesting. Somehow it makes sense. If you have initially higher carrying capacity, you contribute more to the biomass, and you are the most important player in the game. Let's say this comes out naturally at the end of the evolutionary process. And then one can do some approximations and solve this equation in some specific cases. And I skip this, and you can find basically analytically something that is very close to what is observed in numerical simulations. So I will finish with just one observation. So in general, we will not know what are all these interaction parameters. So it will be very hard to actually, no? Okay. To actually detect this transition. So what are actually, how can you get the eigenvalues of a matrix that you don't know? Actually, there is a nice method that has been developed by Mathieu Barbier and colleagues. So these are all statistical physicists that work with this kind of matrices. that allows you to derive what is the most likely matrix, interaction matrix, that you would have if you assign the average interaction coefficient, the distribution of abundances, and that's all. So you assign these two parameters, and then you can compute what is the most likely matrix. And it turns out that this most likely matrix is almost identical to the matrix that has been evolved. So evolution brings you in a state that is somehow, is not random, but it is the most likely given the constraints that are imposed by collective level selection. And this is even capable of tracking this popping out of the eigenvalue. So there is some hope that if we find a way to measure what is the average interaction rate, then we will be able to measure the abundance of species. Then we might try to infer when this transition happens. So when structure starts to be important along an evolutionary process driven by collective level selection. Okay, so let me conclude. So I showed that applying community level selection to complex interacting ecosystems generically will improve the collective function. So there is basically no collective function defined as we did that cannot be selected. The speed of convergence to this collective function depends on many different things. And there is one very specific case where selection is ineffective, and this is when the matrices to start with are antisymmetric. So if you can think about it as a predator-prey system. If I'm trying to select maximal biomass in a predator-prey system, if I increase the biomass of the predator, then I decrease that of the prey. So these kind of systems are impossible so you cannot improve the biomass in the case the matrix is antisymmetric. But apart from that case, you might just need to wait a long, long time. So in general, interactions are made more mutualistic by selection for increased biomass. And in general, you will have that selection at the community level is imprinting a structure on the matrix of interactions. And we still don't know exactly what is really the space of final matrices that we can get to. Actually, it will not always be the same. It will be a set of matrices that have given statistical properties. And which matrix will be exactly will depend on the history of the system. So this is something very exciting that we are starting to explore now. Yeah, and finally, there is some hope that structural changes may be assessed without measuring all interaction parameters. And this is it. Thank you very much.
There's no sort of like conflict, like that conflict you said earlier part of the talk, was it still there?
Yeah. Yes, there is a conflict on a short time scale because the species are competing with one another.
Right.
So on average, the matrix is competitive. But even if you have competitive matrices, abundances will adjust so that in general you will go to an equilibrium. Something I didn't really get into the details is what happens when species go extinct, because it can happen the species go extinct. So that requires actually looking a bit on the details of the equations. So the equations will not be valid forever, because at a certain point, if you get rid of more and more species, then you will need to also reframe the matrix, so the matrix will be reduced in size progressively.
So you don't have migration there in your...
So in the simulations, we add a little bit of migration exactly to avoid this kind of problems, but Yeah, the equations can be written also without migration, but then you have to take care to only consider the entries of the matrix that correspond to species who survived. So that means that you can always write the map from one generation to the other, but if you want to go through many generations, then this is a bit problematic.
So in this case, the selection was artificial in the sense that there's a bottleneck, and then we chose one specific community. But if selection was more more resembling the natural case, say in an example where a microbiome and host interaction, where improved function would just increase your probability of getting selected to the next generation, but it's not a strict bottleneck.
Yeah.
Would you see similar results? Or?
So we didn't try exactly this. What we tried was that we wanted to see whether it was possible to have more than one eigenvalue to pop up out of this bulk of eigenvalues. So what we did was, for instance, to switch between two different selective targets. Let's say in one generation you select for the biomass, in the other simulation you select for, I don't know, being, well, another linear combination of the abundances. So you have like two alternative targets. And it turns out that even if you do this, you actually always have one eigenvalue that comes out. And so if you switch very fast, in the end, you will not have structure. The matrix doesn't manage to really settle on a structure. If the switch is low enough, you will have that one eigenvalue pops out, then you change the target of selection, it will go back in the bulk and then pop out in another direction. So you will have this dynamics. We haven't explored it analytically, but numerically, that's what we observed. We never managed to get, something more exciting in terms of what structure you get. You always get like a low rank structure that is imposed on top of your matrix.
When you are saying that if you look at the final matrix of interactions, and that's going to be similar to, I mean, you can sort of predict it on an average, given that you are selecting for, say, maximizing biomass. Is that going to hold in all situations? Can you always predict what the matrix of interactions is going to evolve to? Or?
Well, let's say you can predict what is the most likely matrix if you know not only what you are selecting for, but also what is like the average feature of this matrix in the end.
So if you want to select for a particular trait rather than maximizing biomass, can you predict what sort of interaction matrix you will end up with?
So I think you probably can, but right now I'm not sure how this, so this alignment of vectors, that is, so the left eigenvalue of the matrix aligns with the carrying capacity, which aligns with the abundances of the equilibrium abundances, then this is probably valid also for other functions than total biomass. But I'm not sure that the three would align in that case. I think you would probably have the two align, and you will not have a perfect alignment on the carrying capacities. So, but I should think about it. I'm not sure we looked at that. So the theory holds for other functions. So you will still have this one eigenvalue that gets out. So how much you can predict the structure? Yeah, I'm not sure.
Thank you, Silvia, for a really stimulating set of lectures.
Thank you, and sorry for this tough one after lunch.